Welcome back to the deep dive. Today, we're jumping into the really fascinating world of microbial genetics, specifically how prokaryotes, these simple organisms, manage their genes so efficiently. It's all about saving energy.
It really is. And the story, interestingly enough, doesn't start in some high-tech lab. It actually starts back in 1910 Mexico with a Canadian scientist, Felix Darel.
Right, he was looking into this disease that was wiping out locusts.
Exactly. And he noticed something strange in his bacterial cultures, these clear spots where the bacteria were just gone. Nothing visible causing it, but they were definitely being destroyed.
And those were the bacteriophages, the bacteria eaters he identified later around 1917. He had this big idea, didn't he? Phage therapy.
He did. He saw them as this potential silver bullet for human diseases. But, well, that initial dream kind of fizzled out, at least in the West.
Why was that?
The bacteria were just too quick. They kept mutating, developing resistance to the phages. And that failure, that rapid adaptation, really highlighted a gap in our knowledge.
We needed to understand how they were doing that, how they were controlling their own systems.
Precisely. We needed to get into the nuts and bolts of their regulation. Which brings us to today's focus. The sources we're looking at detail these ingenious ways bacteria like E. coli turn genes on and off almost instantly.
It's all about responding to their environment and conserving energy.
Absolutely. Now, gene expression can be controlled at several levels, making the RNA, processing it, stability, making the protein, even modifying the protein afterwards. But the big player, the one with the largest impact on what the cell actually does, is transcription.
Controlling whether that initial RNA copy even gets made. Makes sense.
And broadly speaking, bacteria use two main strategies for this. One is what you might call metabolic flexibility or plasticity. Just rapid on-off switching when, say, a new food source appears or disappears.
Quick adaptation.
Yeah. The other strategy is more like a preset program. Think of a virus infecting a cell. It often triggers a cascade. Gene A turns on B, B turns on C in a specific sequence.
Okay, so let's get some basic terms down. Some genes are just always on, right? Doing the essential day-to-day stuff.
Those are the constitutive genes. Your baseline housekeeping functions, making tRNAs, parts of the ribosome, RNA polymerase itself. Stuff the cell always needs.
And everything else.
Those are conditional genes. They're the ones under tight regulation, only switched on when their product is actually needed. That's where the energy saving comes in.
Right. And within those conditional genes, the reason for turning them on or off matters. Is it about breaking something down or building something up?
Exactly. That leads to two main system types. First, inducible systems. These typically handle catabolic pathways breaking stuff down, like sugars.
Like lactose.
Perfect example. The system is turned on by the presence of the substance it needs to break down. That substance is called the inducer. Lactose shows up, the genes to digest it turn on.
Makes intuitive sense. No point making lactose-digesting enzymes if there's no lactose around.
Precisely. Then you have the opposite, repressible systems. These are usually for anabolic pathways building things, like amino acids.
Like tryptophan synthesis.
Yes. Here, the system is turned off when there's enough of the final product already available. That end product acts as a co-repressor. Lots of tryptophan, shut down the tryptophan production line.
Okay, so we have inducible, turn on, and repressible, turn off. How does the cell actually do this? It involves proteins binding to DNA, right?
It does, and there's another key distinction here, how that regulatory protein works. Does it stop things or start things?
Ah, so positive versus negative control.
Exactly. In negative control, the regulator protein is called a repressor. When it binds to the DNA near the gene, it physically blocks transcription. It shuts it off. Remove the repressor and the gene turns on.
So the default is go, and the repressor puts the brakes on.
You got it. Then there's positive control. Here, the regulator protein is an activator. It has to bind to the DNA to actually turn on or significantly boost gene expression, often by helping the RNA polymerase bind better.
So without the activator, the gene is basically off, or at a very low level.
Correct. The activator acts like a green light or maybe a turbocharger.
Okay, this is intricate. But how does the cell signal to these proteins? How does, say, lactose tell a repressor protein to get off the DNA?
It's all about shape. It's a concept called allostery. These signaling molecules, the inducers or co-repressors, we can call them effector molecules, generally bind directly to the regulatory protein, the repressor or activator.
And that binding changes the protein's shape.
Fundamentally, it causes an allosteric transition, a change in its 3D structure, and that new shape changes its ability to bind to the DNA at a specific spot called the regulator protein binding site, or RPBS.
So one shape sticks to the DNA like glue, blocking things. The other shape lets go, opening the way. Or maybe one shape helps polymerase bind, the other doesn't.
Exactly that. Shape determines function. And the classic, the textbook example for putting all this together is the lactose operon in E. coli.
Jacob and Monod, right, 1961.
Yeah.
This is a negative inducible system.
Correct. And they introduced the concept of the operon, this coordinated unit.
Yeah.
You have the structural genes that actually code for the enzymes needed.
For the lac operon, that's lac-Z, lac, and lac-A. Beta-galactosidase, permease, and transadelase.
Right. Then you have the control sites on the DNA itself, the promoter, where RNA polymerase binds, and the operator, which is where the repressor binds. And finally, there's a separate regulator gene, lac-I, that makes the repressor protein.
So walk us through it. What happens when there's no lactose?
Okay, no lactose. The lac-I gene is constitutively expressed at a low level, making the lac repressor protein. This repressor functions as a tetramer, 4 units together, and it binds really tightly to the main operator site, O1, which overlaps the promoter.
Blocking the path for RNA polymerase. So gene's off.
Mostly off, but the sources mentioned there's always a tiny, tiny trickle of expression. A few molecules of the enzymes are still made.
Why? Why leak like that?
That's crucial for actually starting the process when lactose does arrive. You need a little bit of that first enzyme, lactose, lac-Z's product, already present inside the cell.
To do what?
To convert the incoming lactose into a slightly different sugar, allolactose. It turns out allolactose is the actual inducer molecule that interacts with the repressor, not lactose itself.
I see. So that basal level primes the pump. Okay, now lactose is present.
Right. Lactose gets converted to allolactose by that tiny amount of galactosidase. Allolactose then binds to the lac repressor tetramer,
Triggering that allosteric shape change.
Exactly. The repressor changes shape, loses its affinity for the operator DNA, and just falls off.
And the road is clear. RNA polymerase can now bind the promoter and transcribe lac-Z, lac, and lac-A genes on in.
Full speed ahead. And the evidence Jacob and Monod used for this involved some clever genetics with partial diploids bacteria with an extra copy of the lac region on a plasmid.
Merozygotes. This helped figure out what acts locally versus what can travel across the cell, the whole cis versus trans thing.
Precisely. Take the repressor, made by the I gene. If you have a cell with a bad I gene on the chromosome, but a good one, I+ on a plasmid, that good I+ gene makes perfectly functional repressor protein.
And that protein can float over and bind to the operator on the chromosome, right? Even though it wasn't made there.
Exactly. The repressor protein is transacting. It's a diffusible product that can regulate genes anywhere in the cell.
But the operator site, oh, that's different. It's just a piece of DNA.
Right. If you have a mutation in the operator, say OF for constitutive, where the repressor can't bind, well, that mutation only affects the structural genes, ZYA, that are directly downstream on the same piece of DNA.
It can't fix or break the operator on the other copy of the operon.
Correct. The operator is cis-acting. It's a binding site, part of the DNA sequence itself. It only controls the genes physically linked to it. That distinction was key proof.
Okay, but there's another layer to lac regulation. The cell cares about glucose too, right? The glucose effect or catabolic repression.
Yes. E. coli is smart. If glucose, its preferred, easiest to use sugar, is available, it doesn't want to bother firing up the lac operon, even if lactose is also around. Why set up a whole new operation when the easy food is right there?
So how does it enforce that preference? This involves positive control, doesn't it?
It does. For the lac operon to be transcribed really efficiently, another protein complex needs to bind to the promoter region. This is the CKP canopy complex.
CKP is the catabolic activator protein, and CKP is cyclic AMP.
Right. And here's the link. Glucose levels control CKMP levels. When glucose is high, the cell makes less CAMP.
So high glucose means low CMP.
Which means the CKP protein can't find enough CAMP to partner with. So the CAP-CAMP complex doesn't form, or forms at very low levels.
And without that complex binding to the promoter.
Transcription of the lac operon remains very low, even if the repressor is off because lactose is present. CAP-CAMP binding is needed for high-level expression. It acts as an activator.
How does it activate?
It binds to a specific site near the promoter and physically bends the DNA quite sharply, over 90 degrees. This bend is thought to make it much easier for RNA polymerase to bind effectively and initiate transcription.
Wow, so it's like it remodels the landing strip for the polymerase.
That's a great way to put it. So for the lac operon to be fully on, you need 2 conditions. Lactose must be present to get the repressor off. A and D glucose must be absent, so CMP is high, allowing CAP-CAMP to bind and activate.
A 2-factor authentication for gene expression.
Sort of, yeah. It ensures the cell makes the most metabolically sensible choice. And just as a finer point, the repression itself is also a bit more complex. The repressor tetramer actually binds best when it loops the DNA by grabbing the main operator O1 and one of two other weaker operator sites, O2 or O3, further down.
Forming a DNA loop to really clamp things down. Okay, let's switch gears to the other major example, the tryptophan operon, or trap operon.
Right, this is our model for a negative repressible system. It controls an anabolic pathway making tryptophan.
And it's got a double layer of control, doesn't it? Repression and something called attenuation.
It does. It's quite sophisticated. First, you have standard repression. Tryptophan itself acts as the co-repressor.
So when tryptophan levels are high inside the cell,
It binds to the trap repressor protein, which is inactive on its own. This binding activates the repressor, allowing it to bind the trap operator and block transcription initiation. Simple enough, and this provides about a 70-fold reduction in expression.
okay, standard negative repressible control. But what's attenuation?
Attenuation is an additional layer, a fine-tuning mechanism that can knock expression down another 10-fold or so, and it relies completely on the fact that in prokaryotes, transcription and translation happen at the same time in the same place. They're coupled.
The ribosome jumps onto the mRNA while it's still being made by the RNA polymerase.
Exactly. Attenuation happens in a region at the very beginning of the trip mRNA called the leader sequence, TRPL. This leader sequence has some interesting features.
It can fold into different hairpin loops?
Yes, it has 4 regions, 1, 2, 3, 4, that can pair up in different ways. Region 1 can pair with 2, 2 with 3, and 3 with 4. The crucial pairing is between 3 and 4. This forms a structure that acts as a transcription-termination signal, a stop sign for RNA polymerase.
Okay, so 3 pairing with 4 means stop. How does the cell decide whether that happens?
It uses the ribosome as a sensor. The leader sequence also contains a short coding region for a small peptide. And critically, this little peptide sequence includes 2 tryptophan codons right next to each other.
Tryptophan codons are relatively rare.
Yeah, right. So think about what happens if tryptophan levels in the cell are low. The ribosome starts translating this leader peptide.
But when it hits those 2 tryptocodons, it stalls, because there isn't much TRP-tRNA available to bring in the tryptophan.
Precisely. And where it stalls is key. It stalls over region 1. This physically prevents region 1 from pairing with region 2. So region 2 is free to pair with region 3 as soon as region 3 is transcribed.
Okay, so 2 pairs with 3.
And if 2 is paired with 3, region 3 cannot pair with region 4.
Meaning the 34 termination hairpin doesn't form.
Correct. RNA polymerase just keeps chugging along, transcribing the rest of the operon, the genes needed to make more tryptophan.
The cell senses the shortage and overrides the stop signal. Clever. Now, what if tryptophan is abundant?
If tryptophan is plentiful, there's lots of TRP-tRNA around. The ribosome doesn't stall at the trapcodons. It zooms right through region 1 and quickly covers part of region 2.
So it gets in the way of the 23 pairing.
Exactly. By covering region 2, it prevents 2 from pairing with 3. This leaves region 3 free to pair with region 4 as soon as region 4 is transcribed.
And 3 plus 4 equals?
The termination hairpin. This structure forms, signals the RNA polymerase to stop, and transcription terminates prematurely before the structural genes are even reached. Expression is attenuated.
It's amazing. The ribosome's speed, dictated by amino acid availability, directly controls transcription termination hundreds of nucleotides away. That combination of repression, 70x, and attenuation, 10x, gives a huge regulatory range, like 700-fold.
It's incredibly precise control built from these intricate RNA folding patterns and the coupling of transcription and translation.
And even after the mRNA is made, there's still more tuning.
Oh, yes. The cell never stops optimizing. Think back to the lac operon. The LAC-Z, Y, and A genes are on one mRNA transcribed together, but the cell doesn't actually make equal amounts of the 3 proteins.
Right. The sources mention something like 3,000 molecules of Z, 1,500 of Y, and only 600 of A. How?
That's translational control. Even on the same message, the efficiency of initiating translation at each gene's start codon can be different. Maybe the ribosome binding site is better for Z than for A.
Or maybe the mRNA structure itself gets in the way.
That too. Sometimes hairpins within the coding sequence can slow the ribosome down. Plus, different parts of the mRNA might degrade at different rates. It adds another layer of fine-tuning to get the exact protein ratios needed.
And finally, the fastest control mechanism of all. Not waiting for genes to turn on or off, but acting directly on the proteins already made.
That's post-translational control, most often seen as feedback inhibition. This is near instantaneous.
How does it work?
Take our tryptophan example again. If tryptophan levels suddenly shoot up, The excess tryptophan molecules can bind directly to the first enzyme in the tryptophan synthesis pathway, an enzyme called anthranilate synthetase.
And this binding changes the enzyme shape, another allosteric transition.
Exactly. The binding of the end product, tryptophan, to a regulatory site on that first enzyme causes a shape change that instantly shuts down the enzyme's catalytic activity.
Stopping the whole pathway at the source immediately. No waiting for transcription or translation changes.
Right. It's the emergency brake, complementing the longer-term controls of repression and attenuation. It provides immediate response.
So looking back at all this, the inducible lac operon with its positive and negative switches, the repressible tryptophan with that incredible attenuation mechanism, plus translational and feedback controls. It's layer upon layer of regulation.
It really is. What stands out is how these systems achieve such precise control using these very physical interactions, proteins binding DNA, bending DNA, RNA folding into specific shapes, proteins changing shape.
It really makes you think about the sheer structural complexity required for even simple life to function so efficiently. These aren't just abstract concepts, they're physical mechanisms at the molecular level. Bending DNA by 90 degrees, forming loops. It's mechanics dictating metabolism.
It is indeed. The elegance lies in how these specific molecular interactions allow the organism to perfectly match its gene expression to its environment, moment by moment. That coordination is key.
A fantastic deep dive into the efficiency of prokaryotic life. Thank you for joining us.